Hello, everyone. Thank you so much for joining us today for the 9th session in the current season of MOCO seminars. It is my honor to introduce Dr. Carsen Stringer. She trained in applied mathematics and physics in bachelor at University of Petersburg and achieved PhD degree in computational neuroscience at Gatsby Computational Neuroscience Unit in London. Her PhD was about discovering a structure in multi-neuron recording strut network modeling. She has started leading her group at Howard Hughes Medical Institute from 2020. Her lab is focused on two major ideas: understanding how visual computations works and understanding how behaviors are represented in the brain. Carsen, welcome to MPI. We are looking forward to hearing your talk. And please let us know whether people can ask clarification questions between your talk, or what is your preference?
Yeah, definitely. People can ask questions whenever. I have the chat open, so I should be able to see chat messages too, but feel free to interrupt as well.
Yeah, thank you so much.
Cool. Thanks so much for inviting me, and thanks so much for the kind introduction. So today I'm going to talk to you about our analysis technique, Rastermap, and how it can help us understand complex neural activity. I'm going to describe how we use Rastermap to understand specifically experiments that were designed and performed by Lin Zong, a talented postdoc in Marius Pakitzari's lab, and Marius Pakitzari is a collaborator on all of this work. So I'm going to explain these experiments and then go into how exactly analyze how Rastermap can be applied to that data, and then explain how Rastermap works. Okay, so big picture, we're broadly interested in the neural mechanisms that underlie cognition, which includes perception, generalization, memorization, and a variety of other cognitive decisions. So most real-life tasks engage many of these different skills, and so it's difficult to study one skill in isolation. So this cartoon of a foraging mouse is meant to illustrate some of the complexity of an animal exploring a complex world. So the mouse may need to generalize from berry bushes it's seen before and eaten from to tall berry bushes, and it may need to also avoid obstacles or dangers in its complex world. So Lynn and Marius have set up experiments with rich cognitive scenarios along with large-scale neural recordings. So today I'll explain how we analyze these large-scale recordings with the technique I developed called Rastermap. And so like I said before, I'll go into the experiment and then the details of the algorithm. All right, so here's the virtual foraging task that Lynn set up. So we used naturalistic pictures on the walls of this virtual corridor. And so there's a corridor with this image of leaves on the wall, and it's always the same image of leaves. And then there's a corridor with circles on the wall, and it's always the same image of circles. And randomly in the leaves corridor, there is a reward given to the mouse. And then in the circles corridor, there's no reward ever given to the mouse. And so here we can watch a video of the mouse going through the corridor. I'm not sure if you can hear the sound or not.
We can't hear the sound.
Okay, well, all right, so now we're in the leaves corridor, and you'll see the mouse is going to stop and get the reward. So the only thing you would hear is basically there's a tone played in each corridor regardless of the type of corridor, so that basically there's a tone here and not a reward. So the mouse learns that the tone is basically only important in the leaves corridor and not in the circles corridor. And we also introduce a delay there occasionally to try to analyze this behavior more in depth. Okay, so the mouse is running through these two corridors: the leaves corridor, where the mouse is rewarded, the circles corridor, which is unrewarded. And so early in training, the mouse is licks throughout both corridors. So in the leaves corridor, the mouse licks throughout, and in the circles corridor, the mouse licks throughout. And these trials are sorted by the time of the auditory cue here. So this is the purple there. And so after 1 to 2 weeks of training, the mouse learns that in the leaves corridor, it should lick because that's when rewards happen. And in the circles corridor, there's no advantage of licking. And so it very rarely licks in the circles corridor and licks consistently in the leaves corridor. And so we can summarize this behavior with a response rate, where response is considered if the animal licks in the corridor at any time before the sound cue. And then also we look at if it responds before the sound cue and also during a 1 second delay that's introduced later on in training. And so basically the mouse responds in these leaf corridors consistently across trials, but in the circles corridor, it doesn't lick reliably. So it learns that the leaves corridor is rewarding, whereas the circles corridor is not rewarding. So from here, Lynn did a series of tests and further training to probe various aspects of visual cognition. I'm going to specifically focus on the generalization task that he performed, which is asking this question: can mice generalize to new leaves or circles corridors? So are there any questions about the task before I move on to the next step? Okay, cool. I figure this is a more computational audience, maybe. Okay. So the next step is to ask the mice to do visual generalization. So this is the first session where they're going to be shown two new exemplars of leaves and circles. So they're going to be shown these leaves new, which is basically we take this leaves picture and we take new random crops of this image and concatenate it to make a new leaves corridor. And then the circles, which again, we take random crops of this image and concatenate it. And so in this training, these new images were never rewarded in order to prevent the mice from learning about their sort of valence during the session. So there's basically these leaves new and these circles new. And the question is, will the mouse kind of decide that the leaves new are similar to the leaves and that they should lick in both cases, and that the circles new are similar to the circles and that they shouldn't lick them? And so, yeah, sorry.
Did you have training that's for example, silkers be rewarding and leaves not be rewarding? Did you have this case?
Oh, reversal training?
Yeah.
No, we never tried.
For some of the mouses, the circles be rewarding.
Oh, I see what you mean. No, for all the mice so far, it's been the leaves have been the rewarding texture and the circles have been the non-rewarding. But that's a good point. In theory, we expect, well, I'll show the result, but in theory, we hope it holds for other types of images. So the result that we found was that for these leaves textures, the mouse was able to kind of drew this imaginary decision boundary and said that these leaves are sufficiently similar to these leaves and that I should lick during that corridor. And so across 3 mice, we found that the mice licked consistently to the leaves new in a similar way as they did to the leaves, suggesting that the mouse does this generalization. And so this may not be surprising to people who study human cognition, but generally speaking, people don't think of mice as very visual animals. And so it's never been shown before that mice can kind of do this generalization across texture classes. So this was sort of exciting. And similarly, what we would expect is that the circles knew, since they're in kind of the same texture class as the circles, the mouse, 2 of these mice also learned that, or just automatically don't lick to these circles because they don't think that they should be rewarded. And so we have this generalization task where the mouse in this session, first time it sees these images, it thinks that it's kind of in a similar type of place and that it should lick versus not lick in this place. And so fortunately, we also have large scale neural recordings going on at the same time, so we can see what neural populations are doing this task. And so we use 2 photon calcium imaging to record from very large populations of, oh, there's a question, sorry. Do you know if they can at all discriminate between the natural leaves and the cropped leaves image? Well, I'm not going to show this here. So Alex Kipnis asked, do you know they can discriminate between these 2 images? I will show that within 1 session, they start to realize that this isn't rewarded and they stop and they will slow down their licking in this case. And then actually over training, they do learn that this is a distinct leaves. They can learn that as well, and they do. So they realize that this is not being rewarded by us and they stop responding to it. Does that answer your question?
Yes, it does. I was just afraid that perhaps we would interpret something as inductive learning where the mice actually just don't realize that they're in a different corridor.
Yeah, actually, so I'm going to show something a little bit later on, it's a great question. So I think they do realize they're in a different corridor even on this first day, but then they can learn that maybe naturally they're more likely to do this generalization, but then they can learn that this is in fact a place that's not rewarded and it's not the same, and then they will stop looking to this place as well. Yeah, it's a really good question. Okay, so and then to kind of answer your question of do they see it differently, we're going to look in the neural activity. So we use 2-photon calcium imaging to record populations of around 50,000 neurons simultaneously at a rate of 3 hertz. So this microscope is actually a mesoscope, so it allows us to record from this large area around visual cortex and surrounding areas. And so this mesoscope was developed at Janelia, and it was further upgraded at the Allen Institute to record these. You can see these 2 planes here. These are recorded near simultaneously, and here's a little zoom in of a subset of each of those planes. So we can get many, many neurons at a fast rate. And then we extract these neurons using our pipeline, Sweet2P, which lots of other labs do, but I'm not going to go into the details on that today. Okay, so now what do we do with all of this neural activity? Let's make a raster plot. So let's try to plot it somehow. So we have neurons on this axis, and we have time here, and each of these dots is when these neurons are firing. Our proxy for firing, which are these calcium signals that you saw flashing on the previous slide. And then we've kind of put behavioral markers on this plot. So the times of the tones in the yellow, the reward, which is the first lick after the tone, is in green. And then you can see the licks of the mouse are in red. And then the 4 different types of corridors are these 4 different colors, leaves, circles, these ones that they've been trained on, and then these new leaves and new circles images. And so it's nice. We've colored this plot in a way that should be useful, but we can't really see anything because these neurons aren't in any particular order. So we've created an algorithm which puts neurons which have similar activity close to each other. And so this is what kind of this algorithm does. So we have neurons on this axis and time on this axis. And you can now see actually these sequences within each of these corridors of neurons firing to these different corridors as the mouse runs through these corridors. And so there's sequences for the new stimuli as well. And there's lots of other signals going on. So we're going to start zooming into this now that we've made this plot, which I'll go into the details of how we make the plot later on, but we can kind of zoom in and look at different features of this plot. So the first signal we're going to zoom in on are particularly these visually driven populations, which are these populations that go through these sequences. So the mouse enters, for instance, this circles corridor, and these neurons fire in this sequence here. And then when it enters a blue corridor, this sequence fires, this population here, and then this population fires in this sequence, and this population fires in this sequence. And so we're going to pull out each of these sequences. So here's the sequence for the blue leaves corridor. And then this is the sequence for the circles corridor, and then for the leaves new, and then for the circles new. These are different subsets of neurons in our population. And so we basically, you can see that the sequences are only happening in the corridors that are specific that these populations are tuned to. You can see across these different populations. And then in particular, you can see in population 1, these kind of breaks in the sequence, which basically corresponds to the mouse stopping whenever it licks. So it stops moving, and it licks, and it sits there, and there's no kind of visual flow going on past the mouse's eye, and then it starts running again, and then this sequence continues onwards. All right, and now we want to know where are these neurons in the brain that are visually driven during these sequences. And so let's look at population 1, which is driven by these leaves, which are the rewarded stimulus again, and the well-trained stimulus. Okay, and what we see these neurons all over kind of this area, this large 4x4 millimeter area we've recorded. We can overlay brain regions on this recording, and then we can label them as well with the Allen Atlas. And so I'm not going to go into specific details about these different areas because we're not so sure about what all these different areas do, but I'll specifically point out primary visual cortex versus these higher order visual areas. So what you might see is that there's actually quite a few neurons that are visually driven by these sequences in these higher order visual areas, not in primary visual cortex. And for people who don't study mice, there's not kind of this one-to-one mapping between primary visual cortex in mice and V1 in monkeys. But there is sort of, there are higher order areas in mouse visual cortex, these areas around here, which also have retinotopy, which code for what people think of as higher order stimuli. So we have kind of, we can think of this as a primary visual area, like a primary visual area in monkeys, and then these higher order areas as coding for higher order types of stimuli. Although the analogies are not super, they're not super similar. But yeah, for people who aren't so familiar with the mice, just to let you know. Okay, so then we have population 1. If we do look at population 2, we see a similar sort of trend where there's several neurons in this outer part around visual cortex, and there's also neurons responding in visual cortex. But then if we look at population 3 and population 4, these neurons are the neurons driven by these sequences of images are more predominantly found in primary visual areas. And so we think this is sort of consistent with a hypothesis that these well-learned environments are better represented in higher order visual areas. Kind of the mouse has learned their representations, and maybe it activates these higher order visual areas. Whereas in these novel environments, we have more activity in primary visual cortex. Okay.
But
does anyone have any questions?
Sorry, couldn't this higher activity in primary visual cortex also stem from the higher spatial frequency in the cropped images?
So it's not. Let me go back to the images. Sorry. So these are both crops actually. So the way we made these, it's not totally clear that that's the case, but we took a large image of a texture and we took crops and we concatenated them. And that's the case actually in both of these. It might be a little more obvious in this one where we concatenated, but in both cases we concatenated.
Okay.
So the spatial frequency should be the same for both. There are differences in spatial frequencies from leaves versus circles. So that's something we want to test, is if we normalize the spatial frequency, can the mouse still do this task? We suspect so based on passive recordings, but we haven't tested that.
Yes, at least intuitively for the circle environment, it looks to me like the circle's new is more fine-grained than the old circles.
Yeah, I guess it's partly because this is a zoom in. I mean, there's going to be, this is the full corridor.
Fair.
So yeah, so there, when we take the cross, we also zoom in and out a little bit. So there will be parts that are more and less zoomed in, which is something which is to make the corridor more interesting looking, but it can lead to differences in spatial frequency, yeah. But in theory, both should be the same. Okay, there's a question from Charlie Heller. Before training or early in training, do populations 1 and 2 also consist of more V1 neurons? Yes, that's what we've seen, is that it tends to be the case that there are more neurons later in training in higher order visual areas. Yeah, so but we don't know what these representations are, but we suspect they're encoding some higher level features of these corridors or some kind of representation. Okay, so now we're going to kind of ask our question that we care about, which are, what are the neural populations which generalize? And so one way we could do this generalization task actually is to take these visually driven neurons and average their responses. So if I take the average responses of this population 1, which responds during this corridor, it in fact has higher responses to both the leaves corridor and the leaves new corridor. So if you were to average these neurons, you can get a sense of kind of which type of corridor you're in. Likewise, you can do the same thing with the circles corridor. If we average the circles, these responses, we have high responses in these neurons to the circles and the circles new, but not to the leaves and the leaves new. And then, hold on, we have another question. Okay, from Katerina, this is a good question. So was it faster for the mice to stop licking after a number of sessions in the leaves new environment or in the initial circles environment? That's a good question. I'd have to ask Lynn specifically, but my intuition is that the stopping licking in the leaves new environment is faster than learning the task structure to begin with, which is suppressing the licks in the circle environment at the beginning of training. So I think it only takes a few sessions for them to, or even there's one mouse where it really does stop licking during the same session. It realizes, okay, this image is not rewarded, I should stop licking. So I think it is definitely faster in the leaves new environment. But yeah, I'd have to check the data to be 100% sure across all mice.
Thank you for answering. Sorry for the camera off. So that might mean that they also have a sense of what categories are. If it would be faster to unlearn in the leaves new condition.
Yeah, they get this idea. Yeah, they've learned this idea of the structure of the world, that there's a category that gives me rewards and a category that doesn't. And then they can start, if that's kind of what you mean.
Mm-hmm. Thank you very much.
Yeah, it's a good question. Yeah, I mean, because all of these tasks we do with mice are unnatural. So the first thing they have to learn is how to behave in this strange new place where they haven't been before. And that usually is the longest time for training, generally speaking, in a lot of these tasks. Okay. Are there any other questions? Okay. So we could do this sort of averaging, where we sum over these neurons and we get an average over these neurons, which can tell us which type of corridor we're in, whether it's the leaves or the circles. But this isn't necessarily so satisfying. The mouse might need, like, for instance, we expect the mouse maybe has some specific neurons which code for a corridor type where we don't have to average, because the mouse needs to make a decision. And so in theory, there's neurons that maybe, for instance, even do this averaging for us and represent the corridor type, whether it's leaves or circles. And so there's a couple of populations we found that seem to be more like of these generalization type populations. And so one of them I've kind of put in here. It actually gets put on the raster map next to this population. If you zoom in here, you see these neurons that are active at the beginning of the blue corridor, but they're also active at the beginning of the leaves new corridor. So the leaves and the leaves new, and they're not active in the circles and the circles new corridor. So this population encodes the corridor type right at the beginning of the corridor. And so we can take the average responses of these neurons. It actually happens a little before the mouse exactly enters the corridor, because they can see a little bit ahead. And you have this peak in the spike in activity of this population of neurons, or even of single neurons within this population that codes for leaves versus circles overall. Okay, and then there's another interesting signal we found as well, which I'm going to spend a little more time on, which is kind of a sustained generalization signal, where there's this population which is on in both the leaves and the leaves new corridor, but not on in the circles and circles new corridor. All right, and so let's, is it significant? That's a good question. So I'm going to show the novelty. So the novel corridors in general drive a ton of activity in general across the brain. So I think that might be why, on average, many of the neurons are more active during the novel corridor. So that might be, it might be important in that population too. Yeah, it's a good question. Okay, so So we're going to look at this sustained generalization signal. And it's actually a little more complicated than just being on in the leaves corridor and on in the leaves new corridor. Okay, so it is this idea of a generalization signal in that it's not on in the circles and circles. Oh, there's not a circles new here, but it's not on in the circles and circles new corridor, but it's on in both leaves corridors. But it has this special property that it's on, these neurons are on until the mouse gets the reward. So it actually turns off when the mouse gets the reward. So you can see this population turns off here across several of these trials, and it actually stays on throughout the new corridor. And so we can plot this alignment to reward onset. So we're in the leaves corridor. If we plot the activity, we sum the activity of these neurons and sort these trials by when the reward happens. So just to keep in mind, the reward is randomly delivered in the corridor, and so we've sorted by what time the position at which the reward is delivered, and so that's this sorting here. And you can see these neurons are on before the reward comes on, and then they turn off when the reward is after the reward is delivered. And these signals, they're not there in the circles corridor. And just to remind you, so we don't have a reward to sort by, but we have the Q because we always have the tone, no matter what type of corridor it is. So we've sorted by the tone here, and we don't see, these neurons don't care about this tone at all and don't care about kind of this circles corridor. They seem to care about the leaves in particular, and in fact, they also respond in the leaves new corridor, which is not rewarded, but the mouse is generalizing to this corridor. And it doesn't respond in the circles new corridor. And what it looks like is that it doesn't, because we're potentially thinking that because it doesn't get a reward at any point, that these neurons never turn off because they might signal the value of the corridor. And so if I'm a mouse running through this corridor and I get my reward, then the corridor no longer has any value, and so I turn off. But then in the leaves new corridor, I never get my reward, and so this value signal is always on, thinking that at some point I will get a reward, I will get a reward, I will get a reward, but it never actually comes. And over time, in even within this single session, the mouse starts to realize, okay, I'm not getting a reward ever in this corridor, and so it starts to learn, and we can see it in the neural activity, in fact, that over time in this one mouse, this value signal in this leaves new corridor decreases. So these trials now are sorted by time, and at the end of the session, you can see these neurons are less responsive, suggesting that the mouse is starting to learn already that this corridor, although it looks similar to the leaves corridor, is not rewarded. Okay, and then we find these neurons are kind of in higher order visual areas, in particular, these value neurons, these generalization value neurons, and not so much in primary visual areas. Are there any questions about this? Okay, so things that people have been asking about, one of these things is the novelty signals. And so there are neurons that respond regardless of whether it's circles new or leaves new, that respond. You can see these neurons here that seem to code for kind of the novelty of the environment. And so if we make a plot as a function of when the mouse enters the corridor, you can see these neurons are active in the new environments, but not so active in the old environments. And then there's ones that are, so this is a more transient population. This is one that's on for more of the corridor throughout. That's higher during these leaves new and circles new, and less high during the leaves and circles. And these transient and sustained populations these novelty signals seem to be present in primary visual areas. So the mouse, there are signals in the mouse brain that signify to the mouse that this is a new corridor. Nonetheless, the mouse still thinks it's the same kind of context as the leaves original, and so it does lick. But there are clearly neural signals that are distinct during these new corridors. Okay, so we're using this kind of Rastermap technique. We're now able to sort of tease apart these different neural populations associated with cognition So we have these kind of ongoing novelty at corridor onset, and then this generalization / value signal, which is on before the mouse gets the reward, but not after it gets the reward. And then there's also some other populations that code for, like, for instance, when the mouse is in between the corridors, there's this gray screen. There's other populations we don't know what they are. We haven't figured out exactly what they correspond to behaviorally. And then there's this large swath of the Rastermap, which corresponds to neurons, which are more strongly driven by potentially what we think are the spontaneous behaviors of the mouse. So throughout the trial, the mouse is licking and whisking and running at different speeds. And we think that we know from previous work kind of that these neurons can be predicted from the behaviors of the mouse, and you don't necessarily need to know the visual information to predict these neurons. And these spontaneously driven neurons that care about behavior are more kind of in these higher order areas, and there are some in primary visual areas too. Okay, so to sum up kind of this part, we found that in familiar contexts, basically the representations are more in higher order visual areas. We found that there were several neural populations that could do the visual generalization. So for instance, the ones that are driven by the stimulus directly have some generalization signals, but then there's also these kind of special populations in parietal or higher order visual areas that integrate visual category information with the value of the state. So these neurons turn off when the mouse is basically kind of no longer can get a reward. They've already gotten the reward in that context. And so for the rest of the talk, I'm going to talk about how we kind of made this raster map and kind of the more technical details. So I'll pause here, and if there's any questions about kind of the behavioral task and what we did in this first part, what Lynn and Marius did in particular in the behavior.
Hi, Carsen. Can you hear me? Yeah.
Yeah.
So thank you so much for this part. So I have a question regarding these populations which are sort of encoding visual generalization and the population parietal cortex, which is integrating visual category information with the value of the state. So what if you inhibit somehow optogenetically any of these population and try to see the response of the mice in the new leaf corridor?
That's a good question. We haven't done any optogenetic experiments to see what happens.
Okay.
And if higher order areas are more important than primary areas, and so on.
Thank you.
It's a good question, though. We just don't know. There's another question in the chat. So is there any onset time difference between primary visual neuron activity and higher order visual neuron activity? In theory, yes, there is to some extent, but we really can't measure it with these large scale recordings. We're going at 3 Hertz, so we're getting a single sample every 300 milliseconds. So we're not going to get the temporal resolution you would need to see those small differences in onset responses that you would expect from the visual stream. So the Allen Institute has done some really nice experiments teasing apart the temporal relationships between primary and higher order visual areas. So if you're interested in that sort of thing, I recommend looking at their work. There's another question in the chat. Have you tried image style transfer between leaves and circles to see how far the generalization takes place? No, we haven't tried that. And that's something we could try, kind of blurring the lines between the 2 tasks and seeing what happens. But yeah, we haven't tried that. It's tricky because we only really get one chance to do this to some extent, because after the mouse sees that it's not general, I didn't show this, but after the mouse learns the leaves new are different, the next time you show another new leaves, it also says, no, I'm not going to lick to it. So kind of you've broken down this relationship of generalization that might be natural for the mouse to do. And it starts doing this discrimination and has a specific, I mean, you could think of like it knows the landmarks and it's in the corridor that's rewarded, and it's constantly saying, no, this isn't the same corridor. So that's the tricky thing, unfortunately. But yeah, it's a good idea. Alex asks, how many mice models did you use? What do you mean, how many mice did the experiments? Yes, so far this right now is 3 mice have gone through this training, and so a few other types of tasks that I'm alluding to, and Lynn is starting to do more mice with this. So across 3 mice, we've seen this type of generalization, and we've also seen these same types of, I haven't shown this, but these different populations and these relationships between higher order and primary visual cortex are the same across mice as well. So it seems these results seem to be consistent. What happens when the mice see the old leaves again? So they're constantly seeing the old leaves throughout the corridor. So we never try to extinguish the old leaves. So every experiment, they're always seeing the old leaves, and they always are looking to the old leaves. So even though we have the new leaves, which start to be not rewarded, the mouse learns that that's a different type of leaves, and it stops responding to those, but it keeps responding consistently to the old leaves. Yeah, that's a good question. There's another question. So can we say that transient and sustained neurons are complementary to each other? Almost non-overlap? Potentially. I don't know if we have the temporal precision to say that they don't overlap, but they could be sending signals to for different purposes, like setting up a kind of a state change versus keeping yourself in a state that you need to know exactly, like that you need to be on the ready the whole time, versus being like, okay, I don't need to be ready for this corridor. But we don't really know. It's a good question. If one is like one type of attention, another one's a different type of attention. All right. Are there any other questions?
Sorry, Carsen, you have 20 minutes more.
Okay,
yeah.
Cool. All right, so I'll move on. I'm going to go through how we make this raster map, and so how we find kind of structure in this population. I'm going to skip over kind of the motivation for part of raster map, which isn't so important, but basically we find that neural activity is high dimensional, and we're going to actually use this as a constraint to build raster map, but it's not so important that you understand this whole study. So I'm going to skip over it. But just that we're going to constrain our population activity to have this power law decay, but that it is present across different recordings. But let me go, now I'll go step by step in detail into how the algorithm works. Okay, so the first step in raster map is we're going to reduce the data to a manageable size. And so to do that, we're going to use k-means clustering. And so we have maybe 50,000 neurons, and we'll extract 100 to 200 clusters. And the reason we use k-means clustering is because this kind of gives us a partition, kind of covering of the space that hopefully captures all the different patterns that we see in the population. And so we take these k-means clusters, and we take basically all the neurons that are in a single cluster. We can take their average activity, and that gives us a cluster activity for each cluster. So each cluster has it as temporal activity, basically, and we're going to try to extract kind of relationships between these clusters in this temporal space. And so one thing we can do is do a cross correlation. Okay, so, sorry, we have a question. So what's the motivation to use k-means clustering instead of PCA, for instance? So it's a good question. So we want to get different populations in the neural space that are doing that represent different types of temporal information. And so PCA is going to give you kind of an overlapping representations of these different things. And we kind of want to extract out things that are more sparse in the neural space than PCA is going to give us. That's a good question. Shervin asked, how do you decide the number of clusters? And actually, we can't really use more than 200 because of the way I've written the optimization algorithm, which I'll get to in a second. But generally, for the raster maps I've shown you today, those have all actually been run with only 100 clusters. So we have some tricks to run with more clusters, but if your data only has 100 or I mean, it does scale up a little bit, has only so many different types of activity that you need to capture, then that many clusters is okay. So where you might need more clusters is if you're trying to capture really high dimensional responses to many thousands of natural images, that's where we kind of need to bump up this cluster number. But in the case of these recordings, we didn't need to do that. Why would you constrain clustering temporally if your sampling rate is so low? Okay, so why don't I go through how it works? Because I think that's a good question. So we are limited on the sampling rate, but it doesn't mean that we still do have cross correlations that are greater, not at instantaneous time lags, but at various time lags. So if you imagine those sequences through neural space that are happening in our recordings and hippocampal recordings, then you have two clusters that happen in that sequence. They're going to have a cross correlation their cross correlation is going to have a maximum that's not at the instantaneous 0 time lag. It's going to be, for instance, at a 3 or 4 time step time lag, which in our case is a second, and that's okay in the time scale of a mouse running through a corridor, just like these sequences in hippocampus are at that time scale. It's not necessary. It could extract sequences that has faster time scales too, but our data is not fast. Are we sure this is not caused by aliasing? That is a really good question. So in this case, we can run Rastermap on one set of data and hold out the other set of data and check that our sequences are still there on the test set, so that we're not just pulling out noise. You definitely can sort noise, and t-SNE and Rastermap and all these algorithms will definitely sort noise in weird patterns. But that's really only in the case where your recordings are relatively low SNR. So these recordings are pretty high SNR, and we don't. I definitely seen aliasing using Rastermap, but not on these high SNR recordings. So that's a good question. Yes, something to always be careful of. You should always have a test set. Okay, so we have these correlations that we take these time lag correlations across many time bins, and we take the max over positive time lags, and this is so that we capture these correlations in this sequence that can be offset. And so this is pretty important in these recordings that I just showed you to extract this dynamical information across neurons. And so now we get a matrix that looks like this. This is clusters by clusters. This is the max correlation over 10 time lags. And I've sorted, the initial sorting starts as it's sorted by the first PC, and now we're going to try to sort this matrix in a way that we can visualize the global and local structure of the neural activity. And so how do we do that? So we're going to match the neural activity to this matrix, which has a decay, which is a kind of supposed to be a multi-scale matrix. So this decay is quite heavy-tailed. So that the eigenvalues of this matrix has this power law decay. So we're kind of trying to capture that there's this structure that not just clusters that are most correlated to each other should be next to each other, but we want to capture the global relationship between clusters. And then we also have this thing that can be traded off, which is basically we only care about getting local clusters next to each other that are really highly correlated, which is just this off diagonal being one, which is like optimizing kind of this traveling salesman problem. And we kind of can change the weighting between the two of these depending on what we're trying to find in the neural activity. And so we use kind of a weighting of 0.5 to 0.75 for these kinds of recordings with these sequences, and that works fairly well. And so then, as I've alluded to, this kind of this problem, we're constantly multiplying this matrix by the sum of these matrices and maximizing this value. And we're going to maximize it in a greedy way, where you're going to use kind of the tricks that traveling salesman optimizations use. You try to swap nodes, you swap a subsets of nodes of certain lengths, and to try to make the optimization faster, but it still doesn't scale well past around 200 nodes in the way that I've implemented it. But here's what happens while the algorithm runs. And you can see kind of this stuff building up along the diagonal. These are the sequences. And I've kind of zeroed out a lot of the rest of the matrix, so you can't see a lot of the other stuff, but you're going to see kind of building up down here. There's actually quite a bit of off-diagonal stuff down here. These are the neurons that are responsive to kind of these spontaneous behaviors that have more of this global, are more globally active across many neurons. And so we've sorted all of our clusters. And so we have a cluster we have this cluster sorting, and now we need to get back to the neuron space. Okay, does that make sense so far? Okay, so now we have we have clusters, we have maybe 100 to 200 of them, and we can basically upsample maybe a factor of 10 to linear interpolate between the cluster centers in this sorting that we've made. And here I'm interpolating in the PC space. And so there's this interpolated line, and I'm going to assign every neuron now to a position along this interpolated line. And so we have, in this case, I have 150 clusters and an upsampling factor of 10. And this histogram is how many neurons are put in every single position. And after we do this reassignment, we'll get a picture that looks like this, where now we've got something that's neurons by time instead of clusters by time. And so that's how we make this kind of raster plot. Okay, I have more fun examples of raster plots, but I think this is a nice thing to show for people who are working on these kinds of algorithms. There's been recent, maybe I'll call it uproar about t-SNE and its usage, but I think we use these algorithms in order to extract information from these recordings that maybe we couldn't see otherwise. We're not necessarily trying to preserve all of the structure of the recording, but we're trying to preserve some of it. And so kind of the metrics that you're trying to optimize often in these embedding algorithms like t-SNE and UMAP and Rastermap, you're often trying to kind of preserve local neighborhoods. So I'll explain what I mean by this on this toy example. So this is a three-dimensional, this is data that's three-dimensional. Each of these points is a neuron in this three-dimensional space. You can see these groups of neurons that I've colored that are similar to each other. And so if we want an embedding that preserves local neighbors in this high-dimensional, three-dimensional space, I can make a one-dimensional embedding where I put all the blue points next to each other, the red points, the green points, and the yellow points. And I've done a good job of preserving local neighbors. And so hopefully your embedding algorithm does this, and we can quantify this using this is the metric that people often use, which is how many of my original neighbors are still my neighbors in my embedding space. And so I can take, for instance, my top maybe 6 neighbors in this case in my high dimensional space and see how many of them are still my neighbors in my low dimensional embedding space. And so we ask that question for our neural data, and we do that for Rastermap, t-SNE, UMAP, and PCA. We find that Rastermap outperforms t-SNE and UMAP at these local scales of 10 neighbors. And to note, I tried running t-SNE at a variety of perplexities. I tried the multi-scale t-SNE where you run at 2 perplexities and optimize twice, but I wasn't able to outperform Rastermap at these local scores. And so the question, well, I could outperform Rastermap at this local score, but then the global score would be much worse. And so these things always trade off, the local and global scores. So basically, what do I mean by the global score? I look at even more neighbors. I zoom out and look at maybe 100 neighbors. And what you can see is that you can make an embedding that preserves local neighbors, but not global neighbors. So if you take a look at this example, I've got the green points are next to the red points and the yellow points, but really they should be next to the yellow points and the blue points. And so if I want an embedding that it preserves both, maybe it should look more like this. And regardless, I'm always going to lose some information. Like, I'm not going to be able to perfectly assess the fact that some of these green points maybe are closer to these yellow points. I'm always going to lose some of that information. But I can get a better, I'm always going to trade off between these two, but I can try to do as well as possible in the global space as well while trading off. And if we look at our intermediate and global scores, where we look at 100 neighbors and 1,000 neighbors, we see that Rastermap does as well as or outperforms t-SNE, UMAP, and PCA. And so our algorithm quantitatively preserves more local information than these other measures. And generally, I would say if you're looking at neural data, I would not use UMAP. I would use t-SNE, or I mean, I'd say you should use Rastermap, but I'd at least use t-SNE for sure. And also t-SNE and UMAP don't have any way to capture these dynamics that we're capturing with our matrix that with our time lagged correlations. Okay, and so I'm pretty low on time. I'm just gonna show you kind of this idea I was talking about before, where we can take the mouse's face and we can predict neural activity. So this is neural activity, and this is each neuron's replaced by its prediction in this plot here. And this is work that Attica Saida has been doing research on in my lab. And basically, you can compare these two plots. Now that you've got a sorting and a picture of these things, you can kind of look and see where there's discrepancies and what types of dynamics you're capturing kind of in your prediction that maybe you're versus the kinds of dynamics you see in your neural recordings. And you can also look at, because this is a one-dimensional embedding, you can look at the positions of these neurons along this embedding. We can also look at Zebrafish data. So here's a plot of neurons by time in one of these nice recordings from Misha Aaron's lab, where they did a battery of different stimuli that were shown to the Zebrafish, which elicited in some cases strong swimming responses, a variety of different responses. And then we can do the same thing where this is Z-stacks through the Zebrafish brain. We can show where the neurons are in this raster map across the brain, and we can see certain populations that are localized and maybe less localized across the population. You can also use Rastermap on Widefield imaging data. So here's an example of data recorded in Ann Churchland's lab, where they were doing an auditory discrimination task. They also had periods where they had visual stimuli too. And so you can kind of see, for instance, these neurons are visually responsive. This light red is when the visual stimulus is on, and you can see this corresponds to the pink here, which is the pink on this brain. So this might be a way to parcellate. So this algorithm can run also on voxels. It doesn't need to run on neurons, and it might be a way to parcellate wide-field imaging data that doesn't have any sort of constraints. Spatially, things can be put in different places, but generally speaking, it does look like there's some smoothness across space, unsurprisingly, found by the Rastermap sorting. You can also use this on artificial brains. So here's an example on spaceinvaders, where this is a QRDQN that's been trained to play this game. And you can see there's going to be 3 screens that this agent clears before it dies. But basically, it's something we saw in this that might mirror the mice is kind of this value signal decay as the agent clears the screen. And then when the screen gets refreshed, there's lots more value to get as there's more agents to kind of kill. And you can see other types of responses too, and other sorts of sequences that we can find with Rastermap. And here's another example with Sequest. And again, you can kind of see these sequences. This agent isn't actually particularly good at this game, but it tries its best. And then, yeah, I should skip to the end. You can also run it on the Enduro, one that's been pre-trained on Enduro. In this case, there's a huge change in population activity in this period because the agent ends up driving in the snow as well. But yeah, basically you can use this algorithm to look at units in an artificial neural network too, not just in a real brain. And yeah, so I'll wrap up now so there's some time for questions. So we found that Rastermap revealed kind of these cognitive representations in the mouse brain. It captures local structure and dynamical patterns better than t-SNE and UMAP. And we also have this GUI that Attica has built that can help you explore your data and load in behavioral variables and look at what's going on. And so big thanks to Marius Pakitsariou and Lin Zong. And so Lin did all of those nice experiments that I showed you at the beginning, and also my student, Attica Saida. So thanks everyone for listening, and happy to take questions.
Thank you so much for your interesting talk. It seemed very engaging for the audience. And I guess Sebastian has a question.
Yeah, I was wondering, how easy is it to use, like in the sense of how many parameters are there to tune? The traveling salesman versus the other matrix was one, I take it?
Yeah, that's a great question. So that's one that does change the patterns. And so that parameter and the time lag window are kind of the most important ones. So you don't want the time lag window. I mean, at a certain, you can make it a little bit bigger and it's not so sensitive, but you want to make it big enough to capture some of the sequences so that you can get that accurately. But the algorithm is pretty fast to run. So if you're running with only 100 clusters, on my Ubuntu computer, it should take like 14 or 15 seconds on the 50,000 neuron data. So it's pretty fast, assuming you've done PCA first. So it should be fast to test those parameters.
Yeah, and then you kind of verify on this like separate test data, like you said.
Yeah, so there's an option to hold out data in Rastermap when you run it. So you can run it on one part and then test on the other part.
Thank you.
Yeah, that's a good question. Yeah, so it's set up like a, well, maybe that's not an option generally in scikit-learn, but it's set up like a scikit-learn function and with a model.fit and everything.
Thank you. Any other question? Sebastian.
I also had a somewhat naive question, which I should have asked earlier. So you run the k-means clustering on what exactly? That's the PCA data of the neurons, but also in time. So like, yeah. Like
that's a good question. Yeah, we run it on, we take the PCs of the neural activity and we run the clustering on that. So it's neurons by PC to make it faster to run.
Okay, perfect.
Yeah. And we use, so we also try, what generally works better for neural data is to run k-means with the correlation distance, so like a cosine distance, rather than using the Euclidean distance. So that's the default in Rastermap, but you can turn it off.
Very good, thank you. I think people can continue discussions with you in one-to-one meetings after the session. Do you prefer to have a 5 minutes rest or?
Sure, that sounds good. Thank you everyone for coming and for the interesting questions.